Welcome to the Emery Medical Genetics review series. In this series, we make the concepts from Emery Medical Genetics 16th edition, which is the latest edition, clear and connected. Think of it as a roadmap. Instead of reading chapter by chapter, we bring together discussions scattered across the book into one integrated concept, so you see the whole picture at once. These videos are designed for biology undergraduates preparing for advanced studies, medical students mastering their core reference, and graduates or professionals refreshing their knowledge. You can use this series in different ways. Watch first for an overview, learn alongside the textbook, or return later for quick review. Let's dive into today's concept. Hello everybody and welcome back to Instant Review. Today what we'll go through step by step are the central dogma and its exception, different types of DNA sequences in human genome, and finally mitochondrial DNA. Let's get started with the central dogma and RNA-directed DNA synthesis. In previous episodes, we've talked about the central dogma, and we know that it goes back to the idea that a given piece of DNA is made into RNA and then into protein. But it doesn't stop here. Actually, in fact, there are some exceptions where information is converted from RNA to DNA by an enzyme called reverse transcriptase. This RNA-guided DNA synthesis actually happens, and a very good example of this procedure goes back to retroviruses like HIV. But more interestingly, this isn't just limited to viruses. And there is some evidence that even in our own cells, RNA molecules can sometimes act as templates for the production of DNA. And then it can be integrated into the genome. And this is happening in our telomeres by an enzyme called telomerase. Now that we've understand how genetic information flows, the next question is that what is this information written on? Or what does our genome actually consist of? So here we want to speak about different types of DNA sequences in the human genome. Broadly speaking, the human genome can be divided into two main categories, coding DNA and non-coding DNA. So we can take a closer look at nuclear genes. There are about 21,000 protein-coding genes in the human nuclear genome. These genes have some characteristics. For example, they are not evenly distributed across the centromeres. For example, chromosomes 19 and 22 are very gene-rich, whereas chromosomes 4 and 18 have fewer genes and are considered relatively gene-poor. On the other hand, chromosomal regions also vary in gene density. For example, heterochromatin and centromeres are mostly non-coding, but subtelomeric regions usually show the highest gene density. Furthermore, gene sizes also vary greatly, from small genes with only one exon to the biggest known gene, which is TTN gene, with 363 exons. And also, TTNG has the largest single exon in the entire genome. The coding region of the human genome can be divided into two main categories. For example, the first category is consisted of unique single copy genes, and the second category is multigene families. Some genes appear once in the genome. Those genes are called single copy genes and are responsible for the production of essential and vital molecules like enzymes, hormones, receptors, structural proteins, and also regulatory proteins that manage the cell's life function. But during evolution, some single copy genes that were working well undergone duplication and then mutation and divergence. So the result of this process was the formation of gene families or multigene families. Multigene families can be clustered together on a single chromosome, for example, such as alpha and beta globin genes on chromosomes 16 and 11. But also they can be found scattered throughout the genome. For example, we can see the Hox gene family, which is scattered on different chromosomes. not distributed. They are not distributed evenly on a single chromosome, and this type of multigene families are scattered throughout the genome. Multigene families, regardless of their chromosomal distribution, which we had talked about, can be broadly categorized into two main types based on this similarity in their sequences. The first category is classical gene families, and the second category is gene superfamilies. Classical gene families are those families that show high sequence similarity, and they are often found as clustered together, like rRNA and tRNA gene families, which they show striking sequence similarity and are usually found in clusters within the genome. But the second, which were gene superfamily, they have common ancestors, but they have relatively lower similarity in their sequences, and they have diverged extensively in sequence, but they have related functions. The example of gene superfamilies are HLA genes and T cell receptor genes. So here we want to a very major subunit of human genome, which is non-coding segment. It is very interesting that the protein coding genes account for just less than 2% of our entire genome. And this is very, very interesting that the remaining 98% of our genome is consist of non-coding DNA regions, and most of this non-coding segment is consist of repetitive DNA. And here we want to understand different types of repetitive and extragenic DNA. These repeats fall into two principal categories: tandem repeats and interspersed repeats. Tandem repeats are blocks of DNA repeated one after another, and they can be clustered in certain genome regions or scattered throughout the genome. They can be clustered in certain genome regions or scattered throughout the genome. This group these sequences are grouped into three main types: satellite DNA, minisatellite or VNTR, and microsatellites or STR. Satellite DNA, these are the large and highly repetitive arrays, sometimes thousands of base pairs long. A very well-known example of satellite DNA is alpha satellite, which makes up a major structural part of centromeres. Satellite repeats are essential for maintaining chromosomal architecture and proper centromere function, and they represent roughly 10 to 15% of the repetitive DNA in the genome. And the next group is minisatellites or VNTR, or variable number tandem repeats. This group have repeat units of about 10 to 60 base pairs. And a special subgroup of minisatellites is telomeric DNA, which is made of short 6 base repeats at the end of chromosomes. Minisatellites were also used in early DNA fingerprinting because the number of repeats can vary widely between individuals. And the final group of tandem repeats is microsatellites, or STR, or short tandem repeats. This group is the smallest type of tandem repeats, with units only 1 to 6 base pairs long. Microsatellites are scattered all over the genome and are highly polymorphic, which means the number of repeats differ greatly among people. So that's why they are microsatellites are so useful in forensic on the identification, parenting testing, and linkage studies. Despite that most STRs are found in non-coding regions, It has been found that expansion of certain trinucleotide repeats can lead to disease such as Huntington's disease or Fragile X syndrome. Right. Then, the second major category of repetitive DNA is interspersed repeats. This type of repetitive DNA doesn't sit in cluster. Instead, it's spread throughout the genome. And about one third of our genome consists of these sequences. And they mainly come from transposable elements or jumping genes. Interspersed repeats falls into two major classes, lines and signs. Lines or long interspersed nuclear elements make up roughly 5% of the human genome. They are long sequences, up to 6,000 base pairs. And some copies still carry active genes that code for reverse transcriptase, which is then an enzyme that allows them to copy and insert themselves elsewhere in the genome. Common examples of lines include L1 and L2 elements. Because lines can move around and have the ability of um, transcription, lines can sometimes cause mutation or genetic instability. But they have also contributed to evolutionary innovation through gene duplication and diversification. Another group of interspersed repeats is signs or short interspersed nuclear elements. Signs are shorter than lines, around 300 base pairs long, and they make up another 5% of the genome. The most famous example of signs is the ALU elements or ALU elements, which have about 750,000 copies scattered across our DNA. Unlike lines, signs are not non-autonomous, which means that they don't have the ability of move by themselves, and instead they rely on the enzymatic machinery of lines to move around the genome. And finally, we have now become familiar with the various sequences and regions within the nuclear genome. But do you think that it is the end of our story? The answer is not quite yet, because humans also possess another organelle that contains its own DNA, which plays a very crucial role in cellular metabolism, the mitochondrial DNA. Mitochondrial DNA or mtDNA is a distinct type of genetic material which is located in the mitochondria. Each human cell contains several thousand mitochondria, and each mitochondrion carries its own circular and double-stranded DNA molecule with 60.6 kilobases in length. Unlike nuclear DNA, the mitochondrial genome is very compact. It contains very little repetitive DNA, unlike DNA which was consist of about 98% of repetitive DNA. But mitochondrial DNA contains very little repetitive DNA and encodes just 37 genes. These genes include two types of ribosomal RNA or rRNA and 22 transfer RNA or tRNA and 13 protein coding genes. These proteins are essential subunits of enzymes like cytochrome B and cytochrome oxidase, which are critical for oxidative phosphorylation, the process by which mitochondria produce energy for the cell. Interestingly, the genetic code of mitochondrial DNA differs slightly from that of nuclear DNA. So we have different genetic code in some cases between nuclear DNA and mitochondrial DNA, which reflect their unique evolutionary. Another important point is inheritance. The mitochondria present a fertilized zygote come almost entirely from the oocyte or egg cell, which means that mitochondrial DNA is inherited maternally. And so this explains why many mitochondrial disorders follow a maternal inheritance pattern. So, we've taken a journey from the central dogma and its exceptions, through the structure and families of nuclear genes and the hidden world of repetitive DNA. And finally, we talked about the special case of mitochondrial DNA. Congratulations, you've made it! Every video you watch is a step forward in mastering genetics. Keep going, your future expertise is already taking shape.